Hi, my name is Incheol Shin. I'm graduate student at Busan Asian University. Today, I'm going to talk about a protein sequence encoder for secondary structure representation using a pre-trained model. In drug discovery, a protein is one of the most important targets. And this day, machine learning is popularly used for drug discovery. Although proteins are in tertiary structure, they are often represented in primary structure as an amino acid sequence. This representation degrades the performance of drug discovery tools since we lose some real protein structure information to build machine learning models. So, the more structural information of proteins can be expressed, the better drug discovery tools can perform. In this study, we propose a pretrained protein encoder using protein secondary structure. The encoder is inspired from language model, so the encoder has token level task and sentence level task. The encoder's token level tasks are mask language modeling and secondary structure prediction. And the sentence level task is keyword prediction. Our study has three processes: data preparation, tokenization, and pretraining. Data preparation is a process for data pre-processing. We got data from protein data bank. The data have 166,136 proteins, and the format of data is PDBX MLCIF. To use secondary structure for token level task, we got secondary structure from the data using DSSP. And proteins secondary structure is represented by A codes. After getting secondary structure, we made protein words using frequent consecutive subsequence mining called FCS. FCS was proposed in Moore Trends, which was published in Bioinformatics. To use protein structure keywords for sentence level task, we parsed protein structural keywords from MMCI files. And we got 3,190 unique keywords. We didn't have filtered keywords yet, but we will filter the keywords with professionals soon. And next is tokenization. Please see the figure. Firstly, we tokenized protein sequences of our data using the protein words. And then we modified the sequences for tests. For MLM, we masked 50% of all words. That means we changed word token to mask token. And for secondary structure prediction and keyword prediction, we just use tokenized sequences. In pretraining, all tests share the encoder. Our encoder is based on BERT, which is the most representative pretraining model in natural language processing. Anyway, all tests share the encoder, and each test has their own simple multi-layer perceptron for the inference. Mask language model is a test that infers the masked words. And secondary structure prediction and keyword prediction is the same as the name. We benchmark our encoder using TAPE. TAPE's full name is Task Assessing Protein Encoder. We use 4 benchmark data set in TAPE in the result. In secondary structure prediction and remote homology detection, our encoder showed our performance, but in flow scene and stability landscape prediction, our model didn't show our performance. In conclusion, we proposed de novo pretrained protein sequence encoder for secondary structural representation. The encoder showed our performance in secondary structure prediction and remote homology detection, which are tests related to protein structures. In the future, we will focus on physical chemical information. Thank you for listening.